Hello class, this is Mr. Mormon. Welcome to video 7B, a continuation of genetic engineering. In this picture you see Drosophila, the common fruit fly. This is an organism which is very often used in genetic studies. These are the same tiny little fruit flies you'll see flying around the fruit bowl in the summer. There are several advantages to using fruit flies in genetic studies. There are many reasons why the fruit fly makes a good test subject for genetic studies. First of all, thousands of them can be raised in a tiny little area, and they don't eat much. They have hundreds of offspring. Each generation, the individuals will lay hundreds of eggs. And the generations come very quickly. They grow to adulthood quickly and can reproduce, and each generation can be studied generation after generation in a short amount of time. And also the genetics are very simple. They only have 4 chromosomes. Now, one of the things that was learned from studying fruit flies is that Mendel actually got one thing wrong. Mendel, earlier we learned about the independent assortment of allele or genes, the idea that each allele assorted independently and they don't affect one another. But actually that's not true. What we found out by studying fruit flies is that several of the mutations, like you see in this fruit fly here, it has 2 mutations, red eyes and miniaturized wings. These 2 mutations tend to be inherited together. They are linked, and the reason they're linked is because they're on the same chromosome. So it's actually chromosomes that assort independently, not individual genes. Some genes are inherited together because they're found on the same chromosome. In this diagram, we can see gene 1 would assort independently of these other genes, but genes 2, 3, and 4, because they're on the same chromosome, would be inherited together. That's gene linkage. Scientists have been able to make gene maps which show the location of the different genes on the chromosomes. Here's chromosome number 2 of a fruit fly. In fruit flies, the genes are named after the problems abnormal alleles cause, not the normal structures. Like here you see short legs or dumpy wings. So a gene map shows the location of different genes on a chromosome. Okay, so let's talk a little bit about how DNA is studied. The first thing you need to do is extract the DNA or remove the DNA from the cell. So the sample is placed here in a test tube, and detergents or other chemicals are added that disrupt the plasma membrane or the cell membrane, which means basically the cell membrane dissolves and the cells open up, and all the contents of the cell spill out. Chemicals are added that then digest a lot of the parts of the cell, leaving the DNA exposed. If you centrifuge, which means basically spinning this really quickly, the cell components will all separate by density, and all the solid cell parts will go to the bottom. The liquid, called the supernatant at the top, will contain the DNA, which now is no longer in the nucleus, which has been dissolved away. So the DNA is free and floating in the liquid. Adding ethanol makes the DNA all clump together, and it can actually be spooled up on a glass rod and removed. Here's a close-up of some DNA being extracted from a sample of pea cells and a sample of tomato cells, and you can see what that clumped up DNA looks like. You're actually looking at strands of, these would be millions and millions of strands of DNA, and it is able to be seen by the naked eye. Of course, you're not seeing individual pieces of DNA. Those are just molecules that would be way too small to see. But all of the DNA from all the cells will clump together, and you can actually see that. Here the experiment was done with peas or tomatoes, but this could have easily been done with a sample of human cells, maybe saliva, that was a sample of saliva that was taken, which would contain skin cells from the mouth, and you could get DNA that way from a person. What's interesting is this procedure is not something that you necessarily need a a high-tech genetics lab to do. If you do a search online, you will find many kitchen versions of this where you could actually isolate and extract DNA from things like tomatoes, peas, or even your own DNA if you want to see what it might look like by doing this type of procedure. Go ahead, look it up, and maybe this is something you can do on your own. Now, one of the things that often needs to be done is the DNA needs to be cut. That's where restriction enzymes come to play. A restriction enzyme travels along the DNA molecule, and when it finds a certain combination, it will stop and cut the DNA at that location. So restriction enzymes cut at a specific location. And there are different restriction enzymes that cut at different locations. And scientists can use this to do a lot of different things. The DNA can be cut with restriction enzymes, and you end up with all different length segments of DNA. It can then be run through a gel. This is called gel electrophoresis. The molecules of the DNA have to squeeze through the molecules of the gel. An electric current is supplied because DNA has a slightly negative charge. When you place the DNA in a gel like you see here, and you place it in a saltwater bath and apply electricity to it, you put the negative electrode near the well where the DNA is, positive opposite that. The molecules are going to want to move, because they have a negative charge. Opposites attract in electrical charges. So the negative charges want to move away from the negative end and towards the positive end. To do that, they have to go through the gel and pass through the molecules of the gel. The smaller strands of DNA can move more quickly, and they make bands in the gel further down. So down here at the bottom of the gel, you get bands of the smaller segments of DNA, and here we see some larger segments of DNA that didn't travel as far as fast. The DNA bands can then be compared and analyzed, to make discoveries. Here we see a gel with DNA that has been treated with a fluorescent dye so that you can see the bands more easily under black light. They appear very clearly through the gel. Up here at the top of the gel, you can see the wells where the DNA was originally placed. Then when the electricity was turned on, the DNA moved through the gel, creating the bands that you see. Of course, down here at the bottom would be the smaller strands of DNA. They're the ones that went the farthest. These would be much larger strands of DNA, making the bands further up. So here a fluorescent marker was added. Sometimes it's a radioactive marker that's added, and then a photographic or X-ray film is placed on top of the gel so that the bands can be seen. Or the DNA is just simply dyed so that you can see where it ends up in the gel. So what can this be used for? Well, here is some DNA evidence from a crime scene. Some DNA was left behind at the crime scene. Maybe it was a small blood stain, or saliva or semen or any body fluid, or even a few cells or a hair. This is the DNA that was collected at the crime scene. These are 3 possible suspects in the crime. If you were the detective, who would you want to ask some more questions to? Here's a couple other examples. Again, another crime scene. This is the victim's DNA. This is the specimen that was found at the crime scene, and 3 possible suspects. You can see that the DNA of the specimen did not come from the victim, but it does match suspect number 1. That doesn't prove that they committed the crime, but it does prove, they would have to explain why their DNA was found at the crime scene. And here we see gel electrophoresis used to prove paternity. This first column is the DNA of the mother. The next column is the DNA of a child. And these are 2 possible fathers to the child. And you can see by matching it up, in the places where the child has a band that the mother does not, it matches up with father number 1, not father number 2. This proves father number 1 is the true father of the child. Gel electrophoresis can also be used to figure out the actual code, the message on the DNA. This is similar to what we did in the DNA sequencing lab with the where different restriction enzymes are used to cut the DNA in different locations. In addition to using different restriction enzymes, you can add a different fluorescent dye, and then you get a different color combination here telling you the order of the bases in your DNA strand. Okay, that's about it for this video. We're going to have to continue with Genetic 7C because genetic engineering is turning out to be a pretty long topic.